All right, hello everyone, and welcome back to lecture 5 for this week's chapter. This section, we're going to be talking about sexual differences in sexual behavior. So here we have a couple of the main points. Hormones cause sex differences by acting during development as well as in adulthood. There are some sex differences in behavior. There are sex differences in physiology. There are sex differences in disease susceptibility. There are sex differences in neural and glial structure and neural connectivity. There are sex differences in neurochemistry. Androgens and estrogens play a role in sexual differentiation in the brain. Sex chromosome complement contributes to sexual differentiation. Sex differences are context dependent, and sexual differentiation depends on 4 key processes: neurogenesis, cell migration, cell death, and the differentiation of neural circuits. So, hormones have 2 general effects on brain organization. During development, gonadal hormones organize the brain. In adulthood, they activate many sex-specific behaviors, such as mating behaviors. Many behaviors show sex differences, such as a female advantage in verbal fluency tasks when during cognitive testing, whereas there's a male advantage in spatial navigation. Sex differences in behavior are not absolute, and they most likely reflect some of the sex differences in the brain. And it appears as though some of the sexual differentiation is also related to the sex chromosomes. So if we're thinking about this again in a research focus, there's the brain-gender continuum. So although biological sex organs can be described using the binary terms female and male, it is difficult to identify a female or a male brain. So there has been some research on this by Liu and colleagues doing MRI studies, and machine learning was used to investigate sex differences in cortical morphology, and they were able to make some distinctions with 96.77% accuracy. Now, Zhang and colleagues, they used these RS MRI studies in the Chromosome Project, and based on the accuracy of nearly 77.75% of their program, approximately 25% of brains could not be assigned to a specific sex during the neuroimaging concept of androgyny. Or rather, it's showing this neuroimaging concept of androgyny there, where we can't see some of those differences. And in another study by McCarthy and her colleagues, sexual differentiation of any trait must be due to the effects of fundamental processes on brain development, neurogenesis, cell migration, cell death, and differentiation of neural circuits on neuronal differentiation and maturation. Now, although anatomical and MRI studies have shown sex differences in brain structure, it's unclear how these differences relate to the quote-unquote femaleness or maleness of a brain. The Zhang group, they hypothesized that a corresponding androgynous gender role would lead to higher self-esteem and better mental health because individuals identifying as androgynous would be free to act in masculine and in feminine ways without the cultural constraints of gender appropriateness or gender norms. As we think about some of the effects of sex hormones on the brain, sex hormones control differentiation in embryonic gonad tissue into the testes. The influence of the hypothalamus, especially in the preoptic area of the medial hypothalamus, and there's the influence of the neural system regions, especially the amygdala, the prefrontal cortex, and the spinal cord. Hormonal actions on the adult brain are referred to as activating effects, in contrast to the developmental organizing effects. Now, if we think about some of those organizing effects of sex hormones, there's the sexual dimorphism. So differential development of brain areas in the 2 sexes, which are as testosterone and estradiol. Estradiol masculinizes the male brain. So aromatase converts testosterone into estradiol, and the Alpha fetoprotein is less effective in blocking estrogen in humans. Thus, human fetuses of both sexes are exposed to estrogen. A male's testes produce male hormones during the fetal development and androgens. And in the developing rat, androgens are produced during the last week of the fetal development and the first week after birth. So there's this specific timeline of some of the onset of influence that takes place there. And now thinking about this in a clinical context with some of the androgen insensitivity syndrome and androgenital syndrome. After the testes have formed in a male fetus, sexual development then depends on the actions of testicular hormones. So in the case of androgen insensitivity syndrome, individuals with an XY chromosome who are a genetic male fetus, they produce androgen, but the body cannot respond to them. Still, they are responsive to estrogen produced by both the adrenal glands and testes. So the genetic male ends up developing a female phenotype, or kind of the physical expression that we associate with females. Now, with individuals with androgenital syndrome or congenital andrial hyperplasia, the exposure of the female fetus to excessive amounts of androgens or rather, it's caused when there's the exposure of the female fetus to excessive amounts of androgens, and the effects end up varying according to the level of exposure. The organizing effects of the gonadal hormones are more difficult to study in humans, so there's a lot that I think remains to be understood fully. The alpha fetoprotein is known to be less effective in blocking estrogen in humans, so human fetuses of both sexes are exposed to estrogen. And this finding suggests that androgens may play a more central role in sexual differentiation in humans. The direct role of androgens can be seen in studies done by John Money and Ankie Ehrhardt. And I think there will be this clinical focus number 2 in your textbook, where androgen insensitivity syndrome and the androgenital syndrome describe this role. When thinking about activating of sex hormones, the adult female sexual behavior varies across the estrous cycle. So high estrogen levels are associated with sexual receptivity. And in rats, high estrogen is also associated with more dendritic spines on neurons in the hippocampus. In adult males, testosterone levels influence the motivation to seek sexual behavior and the production of copulatory behavior, which I think we talked about in the last lecture or two. In this figure here, this is a comparison of the dendrites of hippocampal pyramidal neurons at high and low levels of estrogen in rats. 4 days. The estrous cycle reveals far fewer dendritic spines in the low period. The activating effects of ovarian hormones can be seen clearly in hippocampal cells. And this figure compares hippocampal pyramidal neurons taken from rats at 2 points in the estrous cycle when estrogen levels are high and when they are low. When estrogen levels are high, more dendritic spines and presumably more synapses emerge. These neural differences during the estrous cycle are all the more remarkable when we consider that the cells in the female hippocampus continually change their connections to the other cells every 4 days throughout the animal's adulthood. The hypothalamus is a critical structure involved in controlling copulatory behaviors in male and female mammals. The ventromedial hypothalamus controls female mating posture. The preoptic area of the medial hypothalamus controls copulatory behavior, but not sexual motivation in males. The amygdala controls sexual motivation in males and possibly in females outside of their estrous cycle. And it's believed that the amygdala probably plays a key role in female sexual motivation, especially among the females of the species, or a species like humans whose sexual activity is not tied only to the fluctuations of ovarian hormones. Studying some of the sexual motivation in mating, So in this experiment, which was done by Dr. Barry Everett, a male rat must press the bar 10 times to gain access to a receptive female who drops them through a trap door. The copulatory behavior of the male rat illustrates mating behavior, whereas the bar pressing for access to the female shows this sexual motivation that's taking place. And so that was how some of this was studied early on. Getting into sexual orientation, sexual identity, and brain organization. So sexual orientation is determined early in development. It's impossible to change. It's influenced by genetics and by epigenetic factors during prenatal brain development. There's no solid evidence that points to any postnatal experience directing sexual orientation, not lifestyle or choice, social learning, things along those lines that in years past, I think people were confused about and were advocating for. So this is some of the most recent science studying this and understanding this. Now, humans have sexual-related differences in the structure of the hypothalamus and amygdala. Several hypothalamic nuclei are 2 to 3 times larger in males than females. So the sexual differentiation of the brain results from the effects of testosterone and is complete by birth. Among gay males and lesbians, variations in epigenetic effects could lead to differences in the architecture and the function of the hypothalamus. Differences in the hypothalamus and heterosexual and gay men suggest that gay men form a third sex neuroanatomically because their hypothalamus differs from both females and heterosexual males, which is just interesting to, again, see the effect of some of these hormones on the neural architecture. Architectural and functional differences in the hypothalamus may form a basis for the spectrum of gender identity. So gender identity, we're talking about an individual's concept of self as male or female or neither or both or otherwise. It's a core aspect of self-identity, and it's usually concordant with the birth assigned sex, although not always. And then as we're thinking about transgender individuals, these are individuals possessing personal characteristics that transcend traditional gender boundaries and corresponding sexual norms. There's the identification with a gender different from that was the sex that was assigned at birth. And there's this strong perception of incongruence between one's sense of self and one's body. And there's also this feeling of estrangement from one's physical sex. So as a result, transgender individuals, they may choose to undergo gender transition to align themselves more closely physically with the internal sense of gender that they experience through social conventions. And they also may express this through the use of different names or pronouns or change the way they dress, or they may seek medical intervention as well. More recent fMRI data shows that transgender persons have a different pattern of cerebral networks mediating self-body perception from cisgender individuals. And that's kind of that default network. And this researcher, Khorramshed and colleagues, found that are cerebral differences in the own body processing networks in transgender females and males compared to cisgender individuals. These differences may be related to a person's sense of body congruence, and the functional change is correlated with an increase in perceptual accuracy in body self-perception. These results strongly suggest that transgenderism is related to the cerebral networks underlying the self-body perception. There are several biological factors that could influence the likelihood of transgender identity. This could be chromosomal abnormalities, polymorphisms of the genes for the estrogen and androgen receptors, abnormal gonadal hormone levels, prenatal exposure to certain anticonvulsants, the immune system activity directed towards the Y chromosome. but I think there's a lot of this that's left to be further understood and explored with future research. It is important to note, though, that the differences in sexual orientation and gender identity, again, I know we had said this before, but they do appear to be the result of prenatal events that end up influencing truly the organization and the function of the brain. So this isn't from postnatal social or environmental experiences that are leading these individuals to identify in this way or experience their sense of self in this way. It is from the prenatal period that all of this really begins. Some of the cognitive influences on sexual behavior. So in addition to the amygdala and hypothalamus, the cortex has a role in controlling sexual behavior. However, this role is poorly understood. Damage to the frontal lobes can result in a loss of inhibition about sexual behavior or a loss in libido or sexual interest. And that is the end of lecture 5. See you all soon for lectures 6 and 7.